Okay, so as we just finished with the first portion of our classification of viruses, let's kind of jump into the second portion, which has to do with how do we classify our viruses based on the host that they infect. And to further understand this, we actually have to play a role of, well, how do they choose? How do they get to decide which host to infect? This is kind of a really very neat mechanism in which, first of all, we get to find out that there is no exempt organism. Pretty much everything, every cell that we know can be infected by a virus, and pretty much everything we've tested has been infected by a virus. So all across all the domains, all across all the kingdoms, and every phylum, et cetera, can be infected by at least one type of virus. For example, even bacteria get infections. In case you were wondering that those guys were the bad guys, no, viruses can infect bacteria and damage them and use them to their will and destroy them as well, right? So how do they do so? Well, there's this specific mechanism that we call complementarity, and I'll try and change it up to make a little bit more sense, in which typically both the virus as well as the actual host have proteins sticking out from their surface. We normally refer to these as surface antigens, a fancy name you've probably been hearing around lately. Now, what these proteins are, sometimes they have a little bit of a sugary coat on them, so they get to be called glycoproteins. They kind of act like handshakes, in which both the virion and the host, as they have these proteins sticking out from them, from their more or less edges, if you will, can recognize each other. And when they don't, when they're not complementary to each other, they just kind of go on by. But whenever they do happen to match at this point in time, like a little secret handshake, they go, okay, I know you, you know me. They let one into the body of the other. So what does this mean? Well, if you look at some of the pictures from the textbook, what you're actually seeing is the virion with its protein coat, with its capsid, and these proteins sticking out from them. And they get fancy little names like spikes and things like that. But those particular proteins are able to recognize the host. And when, again, they come together and are able to be complementary to one another, one will grant entry to the other. So again, like a secret handshake into a cool little club. Now, that secret handshake, like I said, is not necessarily only to one particular club, if you will. Rather, it can actually spread across multiple of them. You see, most of these protein recognitions, most of these antigen interactions occur across all species, across all families, classes, orders, phyla, domains, kingdoms, and so on and so forth. To the point in which a particular virus, sure, can infect a particular animal, but it can also readily infect plants, like you see, for example, from the family Bunyaviridae. Similarly, there's the Corniviridae that we have over there can infect both protists as well as plants. So there's no strict limitation, for example, on where viruses can infect, as long as they have that secret handshake. And so what that secret handshake translates into is, again, the surface, the outside of the virion, the little proteins that it has sticking out, usually can come into contact with the proteins sticking out from the host, like a human cell or a dog cell or a chicken cell, if you will. And when those two come into contact, if they can recognize each other, if they're complementary, if they match, in this case, one will let in the other. In other words, the host will allow the virion in this case to get in. Now, how do they recognize each other? What's that part behind that secret handshake? Well, this has to do with the actual shape of the protein that the particular virion is made out of, something we refer to typically as viral morphology. First of all, let's kind of go back to the scale portion and remind you again how much smaller our viruses are. And so here you have a picture from your textbook kind of indicating the differences between a human cell, like a good old RBC right there. And next to it, you see E. coli kind of dangling off to the side, which is usually a couple of microns in size. That's what we're looking into the labs, right? But then as you kind of keep on zooming down a little bit further, you'll notice that the viruses usually are a couple of dozen to a couple of hundred to sometimes even 1,000 times smaller than even your bacterium. So these guys are not necessarily microscopic, but they're nanoscopic. They're significantly smaller in scale. And for those of you who remember this from the last topic, when we talked about how much DNA is present within these particular organisms, you'll notice that, for example, MS2, the bacteriophage in there that we had declared, only had 3 genes. It only had 3 proteins that it could produce. And its size, as you can see, the scale to even E. coli, it's barely the size of the thickness of a flagellum, for example. So significantly smaller, right? And so from that size, that protein coat gets certain different shapes, some really cool arrangements. And so that protein coat that encompasses the virion, so the external structure that we're talking about itself, is made up of repetitive units. What do I mean by that? Is that typically our virus carries the instruction for 1, 2, maybe 3 proteins that are only designed to form the coat. That's it. Unlike humans that were made up of several hundred thousands of them to make up us, these guys need 2 or 3 of them. And what they do, very similar to the discussion in monomers and polymers, they create repetitive units, lots and lots and lots of the exact same protein. And so those repetitive units are called capsomeres. And so when you have lots and lots of capsomeres put together, you form what you know as a capsid, or the protein coat of a virus. Most of them are structural, so they're meant to just kind of keep the shape of the virus itself. But some of them, as we just finished talking about, participate in some sort of function like enzymatics, like the handshake that we were just talking about earlier. They're involved in recognition, sometimes opening and closing and getting into the cells or the host, and so on and so forth. Mostly again shape, but they also perform some extra functions. So what shapes can they take? Well, one of the cool things is that those capsomeres, those repetitive units of those proteins, more or less can take some really cool shapes. Some of them can look like little tubes in the case of a helical structure. Some of them, when we call them polyhedral or spherical, get more of that kind of blobby, globular kind of shape. They even look like a 20-sided die for those of you who are D&D fans, for example. Some of them just take some really crazy shapes. We usually refer to these as complex. But probably one of my favorites to talk about, and we'll display a couple of examples a little bit later, is the binal shape, which is more or less the shape that the viruses that infect bacteria take shape into. They're usually called phages, or also called bacteriophages is the other name. And in case you haven't noticed, they kind of look like little lunar landers. In case you haven't seen the pretty pictures down there, not only do you have the actual capsid itself that encases their DNA or RNA, but then you see this kind of little tubular structure, kind of little legs floating around from it, right? And so all of these are designed, again, to be able to recognize their host, to attach to it, and to inject literally their nucleic acid into the particular host. So again, part of the classification that we look into these viruses also has to do with their shape, with their proteins, their protein profile that they have outside of them, the little casing, the little shell that they have, which consists of, again, repeated units over and over and over that we call capsomeres that ultimately assemble into one capsid. But that's not all. Sometimes, something that we have heard recently, especially in the media, is that some of the viruses out there, not only do they contain their little protein coat, their capsid, but sometimes they can contain an extra little layer of lipid outside of them, just like we do, phospholipids, right? Now, that lipid gets a fancy name called an envelope. Now, the interesting thing is we had already talked about the fact that virions and viruses don't really carry this. So what gives? How come we're now talking about having a lipid layer outside of it? Well, you see, the lipid layer that some of these viruses can carry is not their own. See, virions, viruses, only have their protein capsid and their nucleic acids inside. That's it. Some of them, however, come with this nice little layer surrounding them like a little cell, like an envelope. Where does that come from? Well, that's kind of a really neat concept. See, remember that the purpose of a virus is to kind of get in, hijack, take over, make more of itself, and then leave. Do it again, basically, rinse and repeat. But every now and then, as it destroys the host cell, as it blows it up, it can grab little pieces of the lipids that the cell was made out of, little pieces of its skin, and then put it on itself, kind of dresses itself up, right? So why is it doing that? Why would it coat itself with this extra layer of lipid? Well, kind of in this concept that I always describe as a wolf in sheep's clothing, by putting on a little layer of lipid that belongs to the original host, it can hide. And so whatever happens is, as the virus infects, takes its little coat, puts it on itself, well, now the immune system of the host won't recognize it as foreign. It'll just assume that it's part of the original host anyway. So there's this kind of devious device from viruses, some, not all, that can steal the envelope from their original host, surround themselves, envelope themselves, if you will, with the particular lipids as a way to hide from the particular host's immune defenses. And so some of those viruses are usually referred to as enveloped. And the ones that don't usually do this are usually called non-enveloped, or sometimes are just referred to as naked viruses as well. So you'll get to hear these terminology every now and then too. So it's a great way to hide, it's a great way to protect itself. And certain viruses, we use that as part of their classification as well. So when we're describing some of these, we'll separate them into enveloped and non-enveloped viruses as well. Now, that being said, well, let me kind of show you a couple more examples, including some of the ones that your textbook provides you, is that depending on whether they have this layer of lipid or not, enveloped or not, in this case, naked or not, if you want to go that route, they'll also be classified, because depending on whether they have this envelope or not, it'll allow them to infect certain hosts. It makes it a little bit easier to kind of get in. And so what you're seeing here is a couple of different examples under the taxonomy or the taxon of order on how you can see variations in single-stranded or double-stranded and multiple pieces, single pieces, so on and so forth. As a matter of fact, if you actually go into your textbook, you can actually see the classification of DNA viruses that we have in there, right? Whether they have double-stranded or single-stranded DNA, whether a negative sense or a positive sense. And then we also have the list of the RNA viruses, which happen to be a little bit more vast than usual. And also on the right-hand side, you can actually see some of the diseases that they can actually cause. So a little bit of a good reference behind them. Now, so far, what we've been trying to do is kind of tell you that, well, based on the shape, based on their nucleic acid, and based on the host, we can classify most viruses. But also, when these three things come together, we really need to compare on how does it actually get to infect. How does it do that? How does that handshake come into play? How does the shape of it, how does that envelope help them get in, and so on and so forth? What we normally call the viral life cycle. Now, for the previous topic F that we've been talking about, when we saw worms, when we saw fungi, when we saw certain parasites and so on and so forth, they all had these kind of life cycles in which they were kind of floating around with a particular toxin or sometimes a vector in there that they would get to the host, infect it. That's where they would go from baby to adult. And then as an adult, they would reproduce and then kind of rinse and repeat and do it again. Viruses don't have that ability. They're not mating. They don't have a sexual portion behind them. And as we saw before, they don't really have offspring per se in the normal way that we'd see it, even though we do call it their next population, their next generation. So when we talk about viruses, we declare them as two separate stages. We talk about their virion stage, the external stage, in which they're being transmitted through some sort of vector, some sort of contact, the air, whatever it is. And then we have this internal stage, that virus stage, in which they manage to get inside the host and then do their damage. With that put together, then we're really going to focus on the internal side. Specifically, well, one, how does it find that host, which we already started talking about, that whole infection concept between that handshake that I was mentioning before, right? Then once it finds the host, well, how does it get in? How does it bring in its content, its nucleic acid inside the host? And even if it does manage to attach and get in, well, how does it really hijack the system? How does it make the cell do its bidding? How does it make the host transform to building more viruses rather than doing its own original job? So that involves the process of taking over and assembling itself, and then ultimately destroying the host itself, blowing it up, a stage that we normally refer to as lysis. Okay, so we're going to kind of cover these one by one, side by side, with specifically one particular example. We're actually going to use the bacteriophage example, that lunar lander shape guide that I was talking about a little bit earlier, that bacteriophage. We're going to look at the life cycle specifically when it relates to bacteria first. We'll go into eukaryotes right after. And so we introduce two terms, what we call a lytic cycle, meaning this is the standard cycle of a virus comes in, gets in, hijacks, blows it up, and does it again. It's making more of itself by lysis. But then we're going to introduce a side note called a lysogenic pathway. So wait until that point. I'll get into that and expand upon it. So there's two stages that I want you to know on how a virus takes over. So let's start with the basic one, the so-called lytic replication, okay? And so remember, once again, it needs to be able to find that host, right? It needs to be able to do that little secret handshake to be able to know that it recognizes itself and then hijack inside. So let's go through all 5 stages first. First of all, they need to find each other, right? There needs to be that point of attachment. So how does that happen? Well, believe it or not, it's kind of random. These handshakes are occurring left and right, and then most of the time they're just kind of being denied. But every now and then, the little secret handshake of protein to protein contact between the virion and the host matches. They're complementary. And then at that point in time, they will allow the entry of the actual virus or the contents of that virus or virion into the particular host. So what does it do to get in? Well, recall that most bacteria, most prokaryotes, have a cell wall. They're just not a regular membrane. Sometimes they have a gram-positive wall, sometimes they have a gram-negative wall, or some variations thereof, right? So whenever viruses, phages, as we give them a name for the bacteria ones, try and infect, they kind of have to overcome all of these things. Enter the story of our little lunar lander over here. Part of those little tail fibers that they contain, part of that little tube that it actually has as part of its structure, is involved in getting in. So not only will it find a point of attachment by doing that whole handshake thing that we're talking about, but those proteins as part of its structure are part of its ability to get in. And what they do is kind of bury little holes and poke holes into the membrane and into the wall while kind of dissolving it and destroying it. In particular, there's an enzyme called lysozyme that is designed to kind of eat it apart. So when this little virus, this little lunar lander attaches itself to the surface of the particular host after having recognizing it, it'll start kind of eating it, poking a little hole into it. And then interestingly enough, this binal shape that you're seeing in there, part of that little tube that you're seeing in there, it will act like a little needle and actually process it through as a way to allow its DNA or RNA to get inside. So that little hollow tube acts like a little syringe and kind of pumps in its content, and then that little DNA will start wreaking the havoc inside the cell itself. Just as a way of kind of showing a little bit of scale, I'm kind of showing you the images from your textbook here, and what you see both a bacteriophage, the ones that infect bacteria, on the left-hand side. This is a little lunar lander hanging out on top of E. coli. And then on the right-hand side, by same scale, mind you, there's a virus infecting, in this case, a human cell. So you can see how much difference of the scale of the cells are, but the viruses themselves roughly remain the same, right? Now, if it attaches itself, if the handshake is successful and it says, welcome to our club, well, what does it do? How does it get that material in? Well, there's some variations, again, between prokaryotes and eukaryotes. Remember, cell wall, that kind of stuff, right? But there's 3 basic established methods on how we describe a virus to get in. Let me show you by pictures, a little bit easier to describe. The first method that is normally referred to as direct penetration has to do specifically with that handshake. Whenever the virion and whenever the host find themselves and go, yep, we're buddies kind of thing, then the actual virion will inject its contents to the inside of the cell, and that content will then hijack the system. We'll talk about that after in a moment. That's one version. A slightly different version occurs when you have envelope viruses. Remember that envelope viruses are the ones that are stealing portions of their host as a way to hide from their environment or from their immune system. Well, because that envelope is the same, one of the things that can happen is that phospholipid bilayer that they're carrying, that envelope that they're carrying, can just usually fuse into the actual membrane of the host, allowing the entire virion to go inside. Once the virion goes inside, then it can open and release its contents within the cell too. So a slightly different variation from the first one. And then the third one, this goes back to our days of topic C when we talked about molecular transport and the so-called endocytosis, in which every now and then, sometimes the virus just gets lucky and gets in by way of either triggering endocytosis or the cell or the host itself kind of taking a little swig of water or a little chunk of food that is trying to get in and accidentally takes the virus inward. So there's several different methods on how the virus can get access to the inside. Now, once it gets access to the inside, though, well, now it needs to wreak havoc. It needs to take over. So in this case, it needs to overcome the host system. Remember that the host is doing its normal thing. It's playing its normal little regular life. It's living, it's growing, it's reproducing, it's making proteins, et cetera, et cetera. Well, the virus needs to come in and stop all of that. It needs to basically hijack the system and then take over to make more of itself. So there's two things that happen. As the blueprint gets put inside the host, whether DNA or RNA, it doesn't really matter at this point in time, one of those proteins that I had mentioned before, that lysozyme, kind of starts melting everything inside the host. It starts destroying every single aspect from the original host itself. The only thing that it usually leaves is the host's machinery. Those things involved in replication, transcription, and translation, all of topic D, for those who remember, right? It leaves those things intact so that it can use them to make more of itself. It destroys everything else of the contents of the cell with the exception of that machinery. And so once it does this, then the DNA or RNA of that particular virion or that virus can now recruit the machinery from that cell, the polymerases, the ribosomes, all those guys involved in that step, and start making copies of itself instead. Now, as it makes the proteins that it needs to make, then those little pieces will usually self-assemble simply because of protein interactions and things like that, and then create a brand new virion. But not just one, several hundreds to sometimes thousands. So kind of let's expand upon this just really quickly. Remember that it needs to find it needs to attach, it needs to gain entry, and once it gains entry, it needs to insert its blueprint, its DNA or RNA. And then the rest of the system needs to kind of hijack the inside of the cell to get rid of all the things that it doesn't need, but keep the cellular machinery. And so regardless of which system you're actually looking at, whether eukaryotic or prokaryotic, so bacteria or animal cells and so on and so forth, because remember, eukaryotes have a nucleus or bacteria have cell walls and things like that, it overcomes these things by using its own proteins. It destroys everything else that gets in the middle of it, and then hijacks or recruits the cellular machinery to make more copies of itself. And so ultimately, this also has to do with what sense they're using, whether positive or negative sense. Sometimes, for example, you're seeing on the image that we have here, on the left-hand side, you have what we call a positive single-stranded RNA, basically fresh and ready to make protein. That guy can get in and start making proteins right off the bat. In certain occasions, a negative sense, remember the ones that require an extra step, can make the positive version of that, make multiple copies of that positive version, and start making proteins a little bit faster. Sometimes it can come with both, for example, in which the positive sense will start making proteins directly, and the negative sense will start making the positive versions of them to make the assembly a little bit faster. So it's this very strong collaboration of just what is inside the virus, recruiting or hijacking the material inside the cell to do its bidding. Now, assuming it does this and makes the proteins that it needs to get out. So what happens then? Well, as more and more of the machinery gets recruited and more proteins are being made, basically more havoc is being wreaked inside the cell, more destruction, more death, and little by little, eventually, the amount of viruses it produces, the amount of progeny, the amount of offspring, if you will overwhelm the body of the cell, and ultimately that same lysozyme that we were talking about earlier will just blow up the contents of the cell from the inside out, releasing all the brand new viruses that were made by that particular host. Now, interestingly enough, what we're going to see in a moment is that, well, specifically, the amount of viruses that it produces also changes from virus to virus, but typically they are in the several dozens, the hundreds, to sometimes even thousands, a number that we normally refer to as burst size. So how many of those get released in one little division, right? One little hijacking. But it also has to do with how fast it can do this. And this is usually referred to as burst time, which how long does it take for one virus to get in, one virion to get in and the virus take over, and ultimately release multiple viruses or multiple progeny. That's referred to as burst time. Now, for those of you who remember your scale, you can always compare a human very, readily, if you will, right? And if you compare a human, for example, remember that it takes us, several dozen to sometimes a little bit longer years to be able to make, one more copy of ourselves. Not too bad. Same thing when you look at animals, you look at a dog, a cat, or whatever. It takes a little bit of time before that happens. When you get a little bit smaller, for example, when you look at bacteria, you'll notice that bacteria do it a little bit faster. It takes a matter of sometimes hours, sometimes even days to do so. Not too bad. But when you look at a virus, most viruses are able to infect and hijack the local machinery in a matter of minutes. And so that means that if you compare it to us, when one virus infects one cell, let's just say, okay, it gets in within about 5 to 10 minutes, starts hijacking the system and makes it make all its proteins. And by the end of just say roughly 20 minutes or so, it will now release 1,000 viruses or virion particles. Well, what happens next? Well, those 1,000 virions will now go and infect 1,000 cells, so 1,000 host cells. Well, then what happens, as you can probably start getting an idea behind that? Well, if it takes 20 minutes to make from 1 to 1,000, 40 minutes later, so 20 minutes after the first round, now you have 1,000,000. That's 1,000,000, right? And then 20 minutes later after that, so a grand total of an hour, that's 1,000,000,000, or a billion, right? So you can see how much faster that burst size becomes a consequence, and how much more dangerous it is, and why typically viral infections happen to affect a little bit faster, especially if they're destroying host cells significantly faster and they're making many more copies of themselves also at the same time, right? So this concept of burst time and burst size are critical factors that we pay attention to when trying to understand how viruses really infect. Now, as I was telling you earlier, this so-called lytic cycle or this lysis cycle is pretty much the standard MO of most viruses. Get in, hijack, make more of themselves, blow it up, rinse and repeat. However, in bacteria, and I'll show you examples for eukaryotes a little bit after, there is a modification of that path. In this case, this is called a lysogenic cycle. So the original was called lytic or lysis. Part 2 is called lysogeny or lysogenic cycle. What is this extra step? Well, it's kind of interesting. Lysogeny in this case is a modification of the original taking over. What actually happens here is that when the virion infects, rather than hijacking and blowing everything up really fast, it actually pauses. It goes into this very interesting kind of resting or dormant stage. We normally call that a prophage stage, or the bacterial version of it. And so this prophage, what it does is its nucleic acid, typically it's DNA in this case, rather than starting to produce more proteins and taking over, instead, it goes and hides. Now, where does it hide though? That's the really neat thing about this concept, is that the lysogenic cycle, the nucleic acid, hides within the host's DNA. In other words, it blends into the genome of the host. In other words, the virus and the host become one, if you want to kind of simplify that. There's plenty of different examples on this, just so you guys know, okay? But here what actually happens is rather than producing all of its proteins and assembling itself and making more, instead it kind of turns off. Like I said, it kind of goes into a little dormant stage. Again, we call that lysogeny. And so it will kind of integrate itself, part of its genome, and just hide there until the right conditions arise. Now, since we're talking about millions of variations of viruses, the term right conditions actually varies greatly. It's not necessarily one particular case, right? It can be anything from timing to food availability to growth conditions and so on and so forth. So it can be a lot of different scenarios there. And so what will happen is that the virus genome will hide inside the host genome, and then eventually it will come back out, a process called induction. It will induce to come out and then resume all that lytic cycle that you're familiar with. Produce more proteins, make more of itself, and then blow up the cell. So why is that a bigger case? Why is that a significant issue? Why is hiding a thing more than anything else? Well, for us in microbiology, that plays a much bigger role when we look at it from the standpoint of a eukaryotic cell. Remember, so far we were just using examples in a bacterium. How is that a bigger deal when we talk about a eukaryote? Well, in terms of infection in the lytic cycle in eukaryotes, it's the same thing. That doesn't really change. But do remember that most bacteria, most prokaryotes have a cell wall that varies, have these extra layers that not all eukaryotes have. Humans, we don't have cell walls, right? Even though plants and fungi do normally have them. So there's some differences there. Also, remember size. Bacterial cells are significantly smaller than most eukaryotic cells. So there's a case of scale or size. And so the other great piece behind it is that envelope system, in which most of these virions that have this envelope around them, if they steal that little piece of skin to hide, will make it a little bit difficult to deal with our own immune systems. So why is that a big deal for eukaryotes? Why is that a problem for us as humans? And so this goes back to the example that we started with when we talked about the very first portion of this lecture. As far as the lytic infection, as far as the lytic cycle behind eukaryotic viruses, it's roughly the same. Recognition, entry, hijacking, make more of itself for its synthesis, and then release. That's pretty much the same thing. The same thing as the lytic cycle in prophages. But the lysogenic cycle that we just talked about in viruses happens also in particular eukaryotes. Just gets a fancier name. Rather than calling it lysogeny, we call it latency. They're pretty much equivalent. Just one works for bacteria, the other one works for eukaryotes. Same concept here. The host, sometimes, rather than being blown up by the virion or by the virus itself integrates itself into the particular host and hides for a small chunk of time. Again, conditions vary from virus to virus, but the same thing happens. The actual virus can hide within the genome of the host, merge itself into the host itself, and hide until it requires to come out, that induction part that we were talking about before. Now, why is that a problem? And why is that cool for the virus? Well, part one, let's go behind the benefit for the virus. Well, one, while it's inside the genome of the host, it doesn't get detected. It's not causing any havoc. It's not destroying anything. It's not harming anything. So the host immune system doesn't really need to go after it. That's part one. Part two is actually kind of funny because remember that most of our cells divide. They make more copies of themselves. That whole cool mitosis and meiosis thing that you all learned, same thing applies. But guess what happens? Every single time one of these latent cells reproduces, well, it also copies the virus as well. Remember, it became part of its genome. So whenever the DNA polymerases come in and make new copies of a cell, well, they actually copy the virus also for free. So the virus is kind of taking advantage of that system. Now, the second portion of why this is a bigger concern has to do with where we started. And this is this concept of what we called oncogenesis. For those who remember the HPV example that I gave you, this has to do with triggering cancer. So what are we going to kind of explain behind this? Now, obviously, we have some expectation that you have your background from previous biology classes or from AMP on how mitosis and meiosis works, right? And also the fact that every now and then, when specifically mitosis doesn't quite go as well as we planned, this so-called abnormal cell division can lead to overgrowth of cells that ultimately harm that same host by either draining the resources that it has around it or not performing the tasks that they're needing to do, like things like organ failure because the cells are no longer doing their jobs, right? So what's the big deal here? Well, for those of you who remember topic D, remember that in order to produce a protein, to build a protein from the process of translation, and then in order to create RNA through the process of transcription, you require starting and ending points. You need where to start and where to end. So for a translation, that was called a start codon and a stop codon. For transcription, that was called a promoter and a terminator, right? Well, that's perfectly fine. That's perfectly the concept that we learned from the central dogma. But what if during latency, during that portion that the little DNA of the virus integrates itself into the host, it happens to shove itself into a place like a promoter? Remember, it's integrating part of itself, of its own DNA into the host. But what if it gets into one of those sequences that is in charge of turning on and turning off transcription and translation? Well, in most cases, if you damage the promoter, well, then you don't get to really produce any protein. No more RNA, no more protein. Simple as that. But what if sometimes it gets close enough to a promoter, then rather than turning off the promoter, it makes it stronger, or sometimes stops it altogether? Now, either you can see two stories coming out from here. If it turns it off, no protein is made. Okay, you can see that as not being too bad. I'll explain why that can be. But what if it turns it on? What if it makes it make more than usual? What if it enhances it, if you will? Well, now whatever protein you're making, now you're making lots more. And again, that may not seem as bad until you compare that particular sequence of DNA when it's involved in cell division. Remember that cell division, both mitosis and meiosis, are tightly regulated processes. There's all these specific steps, there's all these specific checkpoints to make sure that a cell grows when it needs to grow, that it divides when it needs to divide, and that's it. Otherwise, it kind of shuts down. But what if this wonderful little virus comes in, inserts itself close to a promoter, and now makes that promoter go crazy? And what if it does it next to a protein or a piece of DNA or a gene that controls cell division? Well, now you can see the problem. Let's just say that it puts itself next to a protein that is in charge of helping with mitosis. And now it makes it overexpressed. Well, now what's going to happen? More protein means more cell division. And now you start getting the picture that that would lead to things like cancer. Now, what happens if it turns it off instead? Well, what if the protein that it shuts down is part of those checkpoints? What if those proteins were the ones involved in stopping cell division so it doesn't go crazy? And now you just shut it down thanks to that viral infection. So you can see that there's these genes inside our bodies, inside most mammalian cells and most cells altogether, in charge of cell division. And when a virus comes in and goes latent and infects, it disrupts that control. By changing the control of cell division, you can feasibly trigger cancer. And so this is what we call oncogenesis, and the triggers are what we call oncogenes, proto-oncogenes, and tumor suppressor genes, or what I have up there as TSGs. Tumor suppressor genes are proteins in charge of stopping cell division, stopping tumors, tumor suppression, as the name implies. A virus comes in, inserts itself in there, and now it can't suppress the tumor. So now the cell keeps on dividing, aka cancer. In terms of an oncogene or a proto-oncogene, genes involved in cell division. Well, now this guy inserts itself in there, damages that particular code, and now it starts making more of that particular protein. Now, cell division again gets over-triggered, and more cell division takes place. Again, aka cancer. So let's go back to our story of HPV. The human papillomavirus specifically integrates itself more often than not in these particular sequences of cell division. And it'll remain dormant a couple of years, 2, 3, 4 years, 5 years, 10 years sometimes, without causing any harm whatsoever. This is why it can remain largely asymptomatic most of the time. But once induction takes place, one of the things that can happen is as it damages that promoter, it can now trigger cancer. So this is where the true kind of deviousness of a virus comes into play. HPV happens to be one of those versions of these latent viruses that can integrate and damage the host DNA in such a way to trigger cancer. There's other ones that can damage it without really triggering cancer, like HIV, in which the virus can just integrate itself into our certain immune cells and just hide there forever. This is why sometimes the onset of AIDS, specifically from HIV, can take place between 5, 10, 20 years later. That integration, that latency that happens with these viruses has a significant effect long-term, right? So this is why we worry about viruses, just aside from causing disease. Yes, as we are already seeing, there's some severe factors, but those that actually can go latent or lysogenic in the case of bacteria also present a second danger. whenever they kind of try and hide and integrate themselves into another portion of the host themselves, they can cause further damage down the line. So this is where our viruses kind of have this other uglier side to them as far as their infection goes.